Hello, everybody. My name is Nick White. I'm a strain engineer at Ginkgo Bioworks, and there's been a lot of slides going around describing the basics of the coronavirus, and I decided to put a lot of these together and then drastically simplify them to see if I could describe the basics of COVID-19 in about 5 minutes. So, without further ado, here we go. So we're going to start with the genetics, and then we'll work our way from there. So the structure of the coronavirus is essentially a particle with a lipid bilayer, and the four important parts to note are the nucleoprotein, which is involved in the RNA protection, replication, and assembly. I would say probably the second most important protein is the spike protein, which basically attaches to the receptor in the cell that it's going to infect. And then there's a membrane protein and an envelope protein. So in brief, what happens when you get infected with SARS-CoV-2 is the spike protein binds to the receptor ACE2. It then gets, it enters the cell. And then after the membrane fuses, the genome of the virus, which is RNA, basically gets translated by the cell's machinery, the ribosomes, and produce all of these mRNAs, which become proteins, which allow the virus to replicate. So when you look at the phylogenetic analysis of this current SARS-CoV, it's fairly similar to a lot of other bat-derived coronaviruses, which is why one of the current hypotheses is that this is a coronavirus that jumped from bats to humans. So looking at the genome, there's a few important parts. It's about 30,000 base pairs. And two of the main, what they call open reading frames, which means they code for RNA, which eventually codes for an enzyme. So this ORF1A and this ORF1B are directly translated. And then these are involved in helping to translate all of these other mRNA and proteins. So the virus is very efficient with its space. There are a lot of overlapping open reading frames, and normally in cells like bacteria, you have one open reading frame codes for one RNA, codes for one protein, but the virus has a lot of them overlapping, and so it basically makes a really, really big, giant mRNA that then either gets processed or it creates this sort of super protein of everything smushed together, and it requires what's called a protease, which is basically a scissors for proteins to cut everything up and free them. And so this is basically saying the exact same thing. So of the proteins, I'm going to focus on maybe the few most important ones. So NSP1, which starts here, degrades host mRNA, which can be involved in, like interfering RNA. These are the proteases 3 and 5, which are involved in freeing the other proteins once you create that sort of super large protein that's everything stuck together. And then there's a few with interesting name. There's a mystery protein, which I think people are not quite sure what it does. And then Bubble Maker and Bubble Factory, I have no idea why they're called that. But they've done experiments to show that these are involved with the replication and transcription complex. Then there's a helicase, helicases are generally involved in unwinding or unzipping DNA and RNA. There's a polymerase, which is a combination of, it looks like 4 different proteins. And the polymerase is basically what allows to read and replicate the RNA. And then very interesting for an RNA virus, it actually has a proofreading capacity, which is very strange. So viruses live very close to the edge of disaster in the sense that they mutate quickly. And if you mutate too quickly, you'll actually not be able to, you'll sort of lose the ground from underneath you. Most viruses don't have proofreading. Most mammalian cells do. It's very interesting that this virus has proofreading capacity, and this has actually led to some antivirals not being as effective as they otherwise could be. And I'll explain that a little bit later. So transcription and translation coronaviruses, essentially the virus enters the cell and releases the RNA into the cytoplasm. Then the translation of proteins necessary to replicate the genome happens. And then these proteins assist the transcription of the structural proteins. Okay, immunology review. So the immune system is basically a set of walls that any infection has to clear. So the first set are physical barriers like your skin, tears, stomach acid, stuff like that. There's intrinsic stuff. Then there's innate immunity, like natural killer cells, those cells that basically like eat, foreign infection. And then acquired immunity happens where your body is able to basically memorize what certain infection looks like. That takes quite a long time. So essentially what happens is the virus is going to bind to these epithelial cells. And as the infection spreads, it releases cytokines. It eventually spreads to the lymph node, and then this is where the adaptive immune system takes over and starts producing these antibodies. I'll probably delete that slide. So the adaptive immune system is a very complicated system. You could spend an entire class talking about it, but essentially the important takeaways are that antibodies are Y-shaped proteins that bind very tightly and very specifically to what are called antigens. And antigens can be a lot of different things, but they're essentially, you can think of them as like bits of an enzyme or bits of a virus. You can have antibodies for a lot of different things, even other antibodies. And so essentially the adaptive immune system, these B cells will take the antigens from a virus or bacteria and then take it and talk to these other cells. And then you get these memory B cells that say, okay, this is an important antibody, I'm gonna produce a lot of it. So how do we test for coronavirus? The 2 main ones are RNA testing and serological testing. So it's important to remember the difference between specificity and sensitivity. Specificity is a test that is able to capture as many patients with the disease as possible. So you'll be, it'll only test positive for patients with the disease and won't lead to false positive for patients without disease. If you want to test that's hyper sensitive, like it can detect like, 1 RNA molecule in a drop, that would be hypersensitive, but you might end up increasing the amount of false positives. So the RNA testing is basically the first test for any virus. It's the same test we do for like HIV. And the overall workflow is you take the RNA, from a sample, like spit or nasopharyngeal swab, those horrifying things where they stick Q-tips down your nose. You use a protein to convert the RNA to DNA, and then you do something called polymerase chain reaction. Just know that it's an exponential growth of copies of DNA. So think of it as like a runaway nuclear reaction, but just for making a ton of DNA. And then with that, you can either analyze it, you can make it shiny with fluorescence, or you can do next generation sequencing, so DNA sequencing. But there's a lot of requirements. You need to know exactly what the virus genome is. There's sample prep. You need some equipment. You need experts and whatnot. Now, there are alternatives for RNA testing. Some techniques that are what's called isothermal, which basically means you don't need the thermocycler. It does make this step more complicated, but they are cheaper and faster. So there's a lot of different variations on the theme. The other way to test for coronavirus is what's called serological testing, which is a fancy way of saying that you're testing the immune response. So like we said earlier, the adaptive immune system makes these different antibodies. One is IgM, the other is IgG. And IgG is the most abundant type of isoform of antibodies. And the tricky part is that humans have different, Ig just means antibody, response to CoV-2. So these are two different cases, but as you can see, so the RNA, like we said before, that's what's in the viral particles that codes for all the proteins. That remains pretty constant for this one patient and then sort of fluctuates for this other. And then this immune system response is quite light for one, but it's all over the place for the other. So the main takeaway is that the RNA is easy to detect at the beginning of the onset, and will sort of decrease over time, whereas the antibodies are not going to be detectable almost at all until your body has responded to the infection. So RNA first, then antibody response. So serological assays, essentially what they do is you have some sort of target protein and antibody, and if it binds to the target protein, what you can do is you have another antibody that's labeled with an enzyme that does some sort of reaction that produces a color. And so you have antibodies binding on antibodies. This is called a sandwich assay for reasons which are obvious. And then you do this reaction and it makes a pretty color. I mean, the history of molecular biology is making things sticky or shiny. So the pros of this is that it can be quantitative and it's very fast. And it's not too expensive either. The downside is that to develop these tests takes a lot of tech investment, whereas the PCR tests are easy, like a high schooler could design those things. It's not complicated. And then it's also the stability of these tests. It has a shelf life. So a common one is basically a pregnancy test. They also have HIV tests, but basically you have a sample and then it runs across this little pad. It'll be stick to something. You have a place for your controls. And then after 8 minutes, if you see a band, you're pregnant or you have the virus, depending on the type of test. I don't think there's a combined pregnancy HIV test. But so potential therapies for coronavirus, you got two types, preventative and therapeutic. Preventative is vaccine. Therapeutic are things like hydroxychloroquine, different monoclonal antibodies, which you basically purify from like other patients or bacteria. There's some peptides, and then there's antivirals. It's important to note that nothing has been approved yet, which is a fancy way of saying the regulatory agencies have not gotten around to doing anything. There's just sort of suggestions. But the basic approach focuses on supportive care. So in this life cycle of SARS-CoV-2, you can target several different stages of the life cycle to fight off the infection. You could fight off the actual entry, which you can do with monoclonal antibodies, which might disrupt this spike protein from binding to the receptor. There are some antivirals which will mess up with this. Translation is basically exclusive antiviral drugs. A lot of those look like DNA or RNA nucleotides, but they're a little different. And basically when the virus tries to replicate itself, it like screws up. And then you can also try to target these other two. So the virus basically uses the spike protein to fuse its double lipid bilayer with the cells, and that's how it basically injects its RNA. And there has been a study done where they isolated antibodies from recovered patients, and most of them were targeting the spike protein. We're pretty good at neutralizing. So just a quick recap of how vaccines work. They basically leverage the body's ability to remember infections. It's called the adaptive immune system. But memorizing what a virus or bacteria looks like takes a little bit of time, which is why when you get sick, or when the first time you get infected, you do get sick. But if you're ever reinfected, your body's memorization basically responds very quickly to crush whatever happens. So I don't know if you guys remember that old George Carlin bit about how none of the kids in his neighborhood got polio because they swam in the New York River, which is basically raw sewage. So their immune system was tempered in raw sewage. That's kind of why, there's some encouragement for kids to like play in the dirt and not use hand sanitizer all the time. Obviously not now with COVID. Definitely use hand sanitizer. So, but the important thing is that the vaccine allows your body to memorize the infection, but you don't get sick. So there's different types of vaccines, and I'm only going to focus on a few of them. But basically, there's live viruses where you're using a version of the bacteria or virus that has basically been defanged or declawed. There's inactivated, where the viruses have basically been killed with pretty harsh chemicals. You know, when people freak out about viruses, generally they're looking at the like less than 1% mercury. I guarantee you all that stuff is used to kill the virus, and they've tested so that it's in such small amount that it's not going to cause a health issue. So, and then there's nucleic acids. So there haven't been an approved nucleic acid vaccine. This is a relatively new technology, but it has some interesting promise. And then the other one is basically if you take away virus parts, so either parts of the protein, like, you know, you could use the spike protein and try to induce an immune response like that. But a good vaccine is going to be safe, effective, and induce an immune response that persists over time. And then you have all the things like, is it stable and easily distributable? So the vaccine trial process is basically you start with small scale healthy volunteers and focus on safety and learn about dosing. Phase 2 is several hundred volunteers to try to figure out side effects and establish formulation. And then the phase 3 is 100,000 volunteers to see if it actually works. So it's important to remember that the duration of immunity differs with each disease. Things like tetanus, you get a vaccine, you'll never have to worry about it. Things like measles, chickenpox, stuff like that. But as you can see, you know, pertussis, mumps, the common cold, you know, that's why flu vaccines are not very effective, unfortunately. This is basically measuring the amount of antibodies specific for this over time. And unfortunately, a lot of these fade. So how does that relate to coronavirus? Well, in general, coronaviruses, the family, they don't induce long-lived antibody responses. So the common cold is another coronavirus. And as people are pretty familiar with, it's not uncommon to get that, you know, once a year in the wintertime. So this is one showing for SARS-CoV-1, not the current COVID-19. This is the SARS outbreak that happened in Asia a few years back. Basically, those antibodies faded after about a year. So of the vaccine types, the ones to focus on, I would say, are RNA, DNA, recombinant protein, viral vector-based, and live or inactivated viruses. And the time frame it's going to be at least another 6 months, but probably more like a year. So it's important to know, though, that there's about 70 vaccine projects in development. Um, this is a breakdown. I'm going to share these slides, and you can click on the candidates. But of the 70, the hope is that maybe between 5 and 10 look good, and then of those, there's maybe 2 or 3 different ones that finish up. So now let's talk about therapies. So anything that ends with MAB is a monoclonal antibody. So monoclonal antibodies, just to refresh, will basically bind the different parts of the virus or receptor and disrupt them from being able to do what they want to do. Chloroquine hydroxyquinone inhibits viral entry, and there's actually a few different mechanisms. I'm not going to go into all of these. Just know that there's many different types of therapies, and they all act in slightly different ways. So one particular antiviral, remdesivir, like I mentioned before, a lot of antivirals, they look like nucleotides, and they get in the middle of replication, but they basically screw it up. And this is a very interesting pattern, which is called error catastrophe. Basically, that since viruses are so close to their mutation rate is so high anyways, if you just push it up a little bit, they basically they lose cohesion. They can't remember what they're actually coding for, and the virus doesn't die, but you stop the infection. So they did some tests on effectiveness of different antivirals. I'm probably going to skip this slide. Here's a timeline of different treatment recommendations. I will also probably skip this slide. So to end this, basically the most effective things you can do, wear a mask, gloves, glasses, even a scarf helps. I know that for a while the CDC was saying that, and the WHO was saying that masks don't work. They definitely work. It's just that there's been no experiments demonstrating it. But you know, with control groups. You know what else hasn't been demonstrated with control groups? The efficacy of parachutes when jumping out of an airplane. Just think about it logically. If you're covering your mouth and you have something that's catching all of the snot and spit that's filled with virus, even if it's not perfect, every little bit helps. And if you get a lot of people using masks, it helps a lot. And again, it's important to remember that the dose of the virus will affect disease intensity. If you only get a couple bits of virus in your body, that gives your immune system a lot longer to basically mount a defense. Whereas someone sneezes directly in your face and you get hit with 10 billion virus particles, you're going to be in for a rough time. So keep washing your hands, keep social distancing, keep your immune system healthy. I recommend exercising every day, just a short 20 minutes of something. You know, try to eat well, avoid sugar. You can consider intermittent fasting and low carb if you're into that sort of thing. As far as things you can get over the counter, I would highly recommend vitamin D, especially if you live in the Northeast like I do, where it's not very sunny right now. Vitamin C, you can take even at pretty high doses, and it's good. Zinc, try to get some daily activity in the sun. I usually take a walk around the block, obviously all wrapped up. And the most important thing to do is don't panic. So this is from The Hitchhiker's Guide to the Galaxy, which I highly recommend if you haven't already read it. And thank you. I want to thank everyone who made the slides, which I basically condensed. So Ishan, who has actually been coordinating all the presentations at Ginkgo, Naveed, Sudeep, Cam, Lexi, Melody, and if I forgot anyone on the list at Ginkgo, just know that, you know, I greatly appreciate all the work you guys have been doing for explaining this to everyone, and I hope that my simplification, I hope this video is found useful by other people. All right, that's all I got. Thanks, guys.